Very good morning. We are approaching noon, but still it's morning, so a very good morning. I am Dr. Ghosh from the Ross University School of Veterinary Medicine, and I'll be speaking about our work on whole genomic analysis of rotaviruses. Well, diarrhea can be a horrible experience, right? It can be, and even worse, diarrhea can be life-threatening to children and infants. And rotavirus A, now according to the ICTV, we should actually call it as rotavirus species A, known as group A rotaviruses, are one of the major causes of viral diarrhea, severe diarrhea in the young of humans and animals. No concrete data is available on animals. However, according to the WHO estimates, approximately 450,000 to 600,000 children die annually from rotavirus diarrhea, especially in developing countries. I will not go into much details about the basics. It has been already presented by my previous speaker. Rotaviruses have 11 segmented genome. They have 11 segments of double-stranded RNA, which encodes 6 structural and 6 non-structural proteins. And there are 3 mechanisms of genetic diversity, and this includes point mutations, rearrangement, recombination events, and most importantly, as with other segmented RNA viruses, rotaviruses undergo genetic reassortment, and it is regarded as one of the most important mechanisms for high genetic diversity of rotaviruses. So until the 2008, most of the studies on rotaviruses focused on the 2 important genes that encodes the outer capsid protein VP4 and VP7, which forms the basis of the current rotavirus vaccines. However, information on just the VP4 and VP7 gene may not be sufficient to obtain conclusive data on the origin, evolution, genodynamics of rotaviruses, because rotavirus have 11 segments and all the segments are susceptible to the mechanisms of genetic diversity. Things became much easier in 2008 when a rotavirus classification working group was formed, and they devised a whole genome genotyping scheme whereby one can assign genotypes to all the 11 gene segments, and nucleotide sequence identity cutoff value was identified for each of the gene, and you can see several genotypes have been reported. This is a bit of old table. New genotypes have been reported after these 2. And the advantage of this was that because of this genotyping scheme, which was based on the whole genome, we got a uniform platform to work on the whole genomic analysis of rotavirus strain. So in the subsequent slides, I'm going to discuss some of our work on whole genomic analysis of rotavirus strains from different species. And I will just focus on the work that we did on interspecies transmission and reassortment events involving different host species. So let's begin with the horses. Prior to this study, there was a paper in Journal of General Virology where they had actually sequenced the whole genome of few equine rotavirus strains from Europe and Latin America. So we had the genotypic constellation of common equine rotavirus strains, and this prompted us to initiate a study on whole genomic analysis of equine rotaviruses that were detected in Japan. So we sequenced 4 strains for the whole genome. That is, we cracked the complete nucleotide sequences of 4 strains of all the 11 gene segments. And as you can see, 2 of the strains known as BI and CH3 had a genotypic constellation or an overall genetic makeup that was very closely related to those of typical equine rotavirus strains that were reported previously from Europe and Latin America. So we concluded that strains H1 and CH3 are typical equine rotaviruses. Interestingly, another strain HH22, we found that the NSP4 gene was very closely related to bovine rotaviruses, whereas the remaining 10 gene segments were very closely related to typical equine rotaviruses. And even more interestingly, the fourth strain, the strain OH4, all the 11 gene segments were very closely related to typical bovine group A rotavirus strains. So based on this, we came to the conclusion that strain OH4 was perhaps a bovine rotavirus that was transmitted to horses. On the other hand, the strain HH22 exhibited a reassortment event whereby we hypothesized that a bovine rotavirus was transmitted to horses, which was infected with equine rotaviruses. There was a reassortment event, and we got a reassortant strain with the NSP4 from a bovine source and the remaining 10 gene segments from an equine rotavirus source. So there was another sample that was lying in the lab for a long time, and this strain is not from Japan, strain H1. It was a strain that was isolated in Great Britain. It is a very old strain, and very preliminary data was available on strain H1. So we decided to perform whole genomic analysis of strain H1, and surprisingly we found that all the 11 gene segments of strain H1 was closely related to porcine rotaviruses. And based on this, we jumped to the conclusion that strain H1 is a porcine rotavirus strain that was transmitted to horses and caused and was associated with diarrhea in horses. So the important rotavirus genotypes of humans are G1, G2, G3, G4, G9, and G12. So until 2006, G12 was considered as a human genotype. And in 2006, we reported the first G12 genotype from a piglet in eastern India. You can see this is cited in Phil's Virology. And subsequently, we went for the whole genomic analysis of the G12 porcine rotavirus strain. And as you can see here, the VP4, VP6, NSP1, NSP3, and NSP5 genes of this G12 porcine rotavirus strain was very closely related to those of porcine rotaviruses. On the other hand, interestingly, the VP1 clustered with typical human group A rotavirus strains. And the remaining genes, that is the VP2, the VP3, and the NSP2, phylogenetically, they clustered separately from the porcine and the typical human rotaviruses. Therefore, it was very difficult to pinpoint their exact origin. So based on these observations, we jumped to the conclusion that this porcine G12 rotavirus strain may have originated from porcine human reassortment events. Alternatively, the porcine and human rotaviruses might have had a common origin, and with time, these strains diverged, as evident from phylogenetic analysis. So to our knowledge, this is the only report of an animal G12 rotavirus strain that has been molecularly characterized. So in addition to that, we also reported unusual G6P13 rotavirus strain from a piglet in India, and we went for the whole genomic analysis. The remaining part of the work was completed this year and was published. So we found that the VP1, VP3, VP4, NSP1, NSP3, NSP4, and NSP5 genes were closely related to typical porcine rotavirus strains. On the other hand, very interestingly, the VP6 and VP7 genes were closely related to bovine rotavirus strains. And more interestingly, the NSP2 gene clustered very tightly with recent and archival common human rotavirus strains. So based on this, we came to the conclusion that the unusual G6P13 strain might represent a human bovine porcine reassortant rotavirus. We actually had a goat rotavirus strain from Bangladesh. We are globetrotting with rotaviruses. So we had Japan, India, and now we're in Bangladesh. So we had a caprine, a goat rotavirus, group A rotavirus strain that was detected in Bangladesh. And since no information was available on the whole genome of goat rotaviruses, we decided to crack the whole genome. And as expected, all the 11 gene segments appear to share a common origin with ruminants, which was expected because goat is also ruminant. So this represents the first whole genome sequence of a caprine or a goat rotavirus strain. Interestingly, we had also human samples that were collected from the same place where the goat rotavirus was collected, that is Mymensingh in Bangladesh. and we decided to crack the whole genome of two G2P4 human rotavirus strain, which is a very common human VP7, VP4 combination. We found that the VP3 gene of the G2P4 human rotavirus strain was very closely related to that of the goat rotavirus strain that was detected from the same geographical area. On the other hand, we found that the VP1, VP6, and NSP2 genes shared a common ancestry with the goat rotavirus strain. They did not cluster tightly in the phylogenetic trees, but they appeared to share a common ancestry, while the remaining 7 genes were very closely related to typical human G2P4 rotavirus strains. So based on this, we came to the conclusion that a caprine rotavirus might have been transmitted to human. It underwent reassortment events with human rotaviruses, generating a caprine human reassortant strain. Subsequently, we jumped from Bangladesh now to Kenya. And in Kenya, we had a human sample B12, and we cracked the whole genome of the human sample B12. And we found that all the 11 gene segments of strain B12 was closely related to the ruminant or bovine rotavirus strains. And therefore, we came to the conclusion that strain B12 is actually a bovine or ruminant rotavirus that was transmitted from animals to humans. And since the sample was collected from a village, so where humans come in close proximity to animals, this may not be unusual. From Kenya, we jumped to Thailand, and we had 2 unusual human rotavirus strains from Thailand, MC323 and MC345, and we cracked the whole genome of both the strains, and we found that most of the genes of MC323 and almost the 10 gene segments other than the VP1 of MC345 were closely related to porcine rotavirus strain. Phylogenetically, we could not ascertain the origin of VP1 and NSP5 of MC323 and VP1 of MC345. And therefore, we came to the conclusion that MC323 and MC345 were perhaps porcine rotaviruses that were transmitted to humans in Thailand. And again, we jump back to Kenya. And there was a very interesting rotavirus strain with the G3P2 genotype constellation, and we decided to crack the whole genome of this strain. Very interestingly, the VP2, VP3, VP4, NSP1, the NSP3, NSP2, VP7, and NSP5 genes of this strain was very closely related to a simian rotavirus strain, SA11, that was detected from South Africa way back in 1958. So this was very interesting. Subsequently, the VP1, the VP6, and the NSP4, after we blasted, we performed homology search, they exhibited nucleotide sequence identity cutoff values which were much less than that is required to assign a genotype. So we submitted the sequences to the RCWG, and they assigned novel genotypes to VP1, VP6, and NSP4. So based on our conclusion that at least 8 gene segments were very closely related to the monkey rotavirus SA11, which was also detected from Africa, while the other 3 genes were not related to any rotavirus gene sequence available in the database based on the RCWG classification scheme, we hypothesized that this may be a simian rotavirus strain or a rotavirus strain from wildlife that was transmitted to human. And this is not unusual because this sample was collected from a village that was located next to the Nakuru National Park. And Nakuru National Park has a sizable and versatile simian population. So it might be possible that the villagers go to the river for drinking, and there was also a bore well where the villagers go for drinking, and the animals also come down for drinking water from the same source, which might have resulted in the interspecies transmission event. However, the child from where the virus was recovered, this child was asymptomatic. And again, we jump back to Thailand, and we detected a G8P10 rotavirus strain, which is again an unusual human GP combination, and we went for the whole genome analysis, and we found that at least the VP1, VP2, VP3, the VP6, the NSP2, VP7, and NSP4 genes were closely related to bovine rotavirus strains, whereas the NSP1, NSP3, and NSP5 genes were closely related to human rotavirus strains. The origin of the VP4 gene could not be ascertained properly. So based on this, we came to the conclusion that the strain 69M, which is a very archival strain, was a bovine rotavirus that was transmitted to human, where it underwent reassortment events with human rotaviruses, and we got a bovine-human reassortant rotavirus. In addition to this, we also worked on human group A rotaviruses that actually had evidence for origin with canine and feline rotaviruses. These are two rare human rotavirus strains that we cracked from China, and by whole genomic analysis, we found that they were perhaps derived from feline and canine rotaviruses. And we also characterized some G2P4 rotavirus strains from Kenya, and we found evidence that they shared a common origin with bovine rotaviruses. And also these are some of the archival P6 strains. We characterized the whole genome, and at least one of the strain, that is 1076, we found that the VP1, VP3, VP6, and NSP4 genes were derived from bovine rotaviruses. 1076 was actually detected in Sweden. And more recent, we also published another paper on strain R479, which was a G4P6 rotavirus from human. But by whole genomic analysis, we found that it is a porcine bovine reassortant rotavirus that was actually transmitted to humans. So now with the advent of the next-gen sequencing, that is the next generation sequencing, more rotavirus strains have been cracked for the whole genome, and more interesting data on interspecies transmission and reassortment events involving rotavirus strains from different host species are emerging. So this is my university, the Ross University School of Veterinary Medicine, and this is the other place where a part of most of the work was done. That is the Department of Hygiene, Sapporo Medical University. So thank you so much for your kind attention.